Welcome to Chapter 1. This chapter is basically intended to be kind of a review of cell biology to kind of refresh what you should have learned in intro biology and to help get us going and moving toward learning more about cell biology this semester. So what should we be doing in this chapter? I added this slide of chapter objectives to give you a brief overview of what I really intend for you to get out of this material. So for example, I want you to get a big picture of where we're going in this chapter and what I want you to learn out of this chapter. And I'm normally a big picture kind of person, but cell biology is totally the reverse. It's really focused on the minute details, and yet within that there's still a lot of different ways that things link together and integrate with each other, and this is what makes our cell operate. So this is kind of what cell biology is all about, and that's part of the reason I love it, is that it's so intricate and it's so complicated. There's so much to learn, and it's this mysterious little world inside the cell, and I really love that. I want to point out something. You'll see that we're going to list and describe the 9 basic properties of all cells. List and describe are 2 different things. List means, yes, it has this, 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 and this. That is not going to get you full credit on an exam question or in an essay or anywhere else in your biology career. Describing them, how, in what way, how does this work? Tell me everything you know about those 9 basic properties of cells and how an object meets those or fails to meet that. So if I ask you, this is a picture of something, is it a cell? You tell me why or why you do not think that it is a cell. The 3rd section that we'll be dealing with in this particular chapter looks at the structure and function of viruses, how viral infections work a little bit, and how viroids differ from full viruses. So this is where we start even early on in the semester getting to talk about some of the medical aspects of cell biology. Why do I like cell biology so much? I've always wanted to know how stuff works. It fascinates me. I love to take things apart as a kid, and this is really the ultimate taking things apart. Cell biology is reductionist, and that's what this means, is we're going to take things apart into their littlest pieces, try to figure out how those work, and then we can start putting them back together to see what those pieces do and how they interact with each other. And that's really what this field of cell biology is all about. How do we understand what the little intricate bits of our cell do, and then how do they work with each other to make us function? During the semester, you're going to be doing a lot of finger twitching, so this is a very good skill for you to practice. Hold up your finger. Twitch. Okay, how does that happen? Because we take this totally for granted, but there's a huge amount of biology and chemistry that drives the simple contraction of a finger muscle on command. So twitch your finger and feel some wonder. And by the end of the semester, your brain's going to melt and you're going to wonder how in the world you ever got away without twitching your finger. But you'll have a much deeper and more intricate understanding of how the cells actually communicate from your brain all the way down to your finger to make those muscles contract and for you to bend your finger, and even what the skin is doing and stuff. It's pretty amazing. So other things that we get to look at include, why do some drugs make you sick, like anti-malarials? If we take them, we get sick, but if we take antibacterial drugs, antibiotics, not so much. What's the difference? Why do some drugs affect us and others do not? This is another part of what we get to look at. The origin of life and the origin of cells is kind of tied together. So this is such a huge field. There's so many different things going on that I can't even begin to tell you all of it in one semester. It would take years and years and years, and I still wouldn't be able to tell you everything. That's why we're still doing research in this field. But trying to understand how our cells work enables us to do things like to develop new drugs that don't make us sick, to target specific disease pathologies so that maybe we can get really good treatments for arthritis, how we understand development so we can correct birth defects or maybe even reverse genetic diseases before they affect children. These are some of the goals and some of the purposes behind cell biology. So the beginnings of cells, you've probably heard the story before. This came about after Robert Hooke developed the first microscope, and that's the one that you see on the left there, that little thing that looks like a screw with a little pinhole in it. Yeah, What you actually did was you took this little thing, you mounted your specimen on the tip of that screw, the little sharp point, and then that little hole, you squinted through it, and you held it up to the light, you squinted really, really hard, and you adjusted that screw until your specimen was just in the path of the light. And this got you your first levels of magnification. Not very good. I'm sure the picture quality was really, really bad, but similar structures and things are still actually used in some field biology today, which is kind of surprising. I think that's changing a little bit because there's now actually attachments that you can put on your cell phone, and everybody has one. So those are actually really revolutionizing our current field of cell biology. The next microscopes that were refined a little bit by Anton van Leeuwenhoek are the more decorative, prettier versions that look something more like what we see today. And these are gorgeous. I mean, look at the thing. It's a piece of artwork. It's not just a tool. I really feel like a lot of scientific instruments today have lost their soul in a sense. We don't make them for beauty anymore, we make them for function. So everything's black or gray and plastic and metal and just kind of boring looking, as opposed to that, which would be a delight. The compound microscope, having multiple sets of lenses, gets you even better magnification. And when we first started to look at structures under these microscopes, the story is that van Leeuwenhoek was trying to figure out why corks kept air in bottles so well. So he looked at a really super thin slice of cork under the microscope, and that's what you're actually seeing in that little inset, the little round bubble there. Those are the exterior structures of what used to be cells, and he called them cells because they reminded him of the cells in a monastery, where monks live and pray and all that stuff. So the modern cell theory is still just a theory. I mean, I hate that phrase, just a theory, because it has so much support and so much evidence. Every living thing that we know of today is composed of one or more cells. So it's our basic unit of life. It is the smallest individual thing that we consider to be functionally alive at this point, and all living things come from pre-existing living things. This is the basic tenets of our cell theory. That may seem really self-explanatory, but as we're out there kind of exploring the universe, I have to wonder if there are life forms out there that don't correspond to our ideas of cells, that don't have this kind of structure that we think of. And what would that look like? Would we be able to recognize it? Is it possible? Yes. Is it probable? I don't know. But it's a fun thing to think about. So what you're looking at in this particular image are some HeLa cells, and these are human cancer cell lineages. These are single individual cells that you can see in outline. The nucleus is stained red and the actin filaments are stained in yellow in this particular case. So cells are the smallest unit of life. You hear that all the time. What we mean is that, like I said in the last slide, they are the smallest entity that we consider to be functionally alive. Most types of cells can actually be grown in culture, which we do scientifically a lot, and that includes things like neurons, fibroblasts in your skin, kidney cells, bladder cells, but some of them have really strict growth requirements. One of the first cell lineages, actually the story comes from here in Virginia, and these are the HeLa cells. They were derived from a cancer patient, Mrs. Henrietta Lacks, who was from Roanoke, Virginia. And her doctor, George Guy, isolated these cells that he was able to keep growing indefinitely in culture. And he was really, really excited about this because it was the first time anybody had been able to do this. Normally, if I were to just take a chunk of skin and stick it in a petri dish, I could keep a piece of skin alive for maybe 5 to 7 days. That's pushing it, but maybe 5 to 7 days. If I were to try to isolate individual cells out of that, I could keep them growing in culture for a few weeks to a few months, maybe even a couple years if they were cared for properly. But eventually they all stop replicating. This is a normal part of our life. But these cells were different. They didn't stop replicating. And so he was fascinated. They ended up being a fantastic research tool, and this guy distributed them all around the world without asking permission. Henrietta Lacks was a black woman. This was in 1951. No vote, no rights to speak of at that point. Segregation was still a thing. It never even occurred to anyone that we should ask permission for something like that. Today, we've gotten a little more strict about it. The HeLa cells have been in the news a fair amount in the last couple years. And there's more to the story of Henrietta Lacks. There's actually been a book published about it. Some of you may have read it by now. It's The Immortal Life of Henrietta Lacks, and it's about the story of her and her cells and her cancer and her struggle and her family's struggle against this part of her body essentially being used for scientific research purposes without any permission or any ethical considerations. Today, her family has kind of accepted that the genie's out of the bottle and there's not much they can do with it. So they are letting researchers continue to do research with HeLa cells. I've worked with them. But before you publish any potentially personal information about those, like genome stuff, you're supposed to request permission from the family to publish any data like that. That's not always followed. In the 1980s, the full medical history of Ms. Lacks was released without her family's permission. She did die of cancer. And in 2013, the full genome of the strain of HeLa cells was published also without the family's permission. So there's a lot of ethical concerns that have come up about this. I've donated cells for research purposes. It doesn't bother me at all. I don't expect anyone to contact me to ask for permission to publish based on my cells. I gave them freely, and yeah. Currently, you cannot patent a human gene, so that at least is something that we have kind of resolved. Nobody thought that was an ethical behavior. So our 9 basic properties of cells, we're going to work through this. Most of these you've heard before. Again, this is really kind of supposed to be review at this stage. You've seen or heard the criteria for life before, and these are basically the same ideas. If you've been through my evolution class, we argue whether horses or Harley-Davidsons are alive. This goes back to the same kind of argument. So what do things have to be considered alive? They have to be complex and organized. They have to have a genetic program. Today we consider that DNA and some sort of mechanism for using that DNA. It's possible there might be other options. They have to be capable of replicating, of reproducing themselves. They have to be able to pass on their genetics to offspring. They need to be able to acquire and utilize energy. This means some sort of basic metabolism. Every living thing that I can think of uses glucose. That may not be true on other planets. It may not even be true in some deep sea vents or for some really bizarre archaea, but glucose seems to be the going fuel. They carry out a huge variety of biochemical activities. We're going to look at all of these in more detail over the next few slides. They engage in mechanical activities, so they have the ability to do stuff. They can respond to stimuli if you poke it. They're capable of homeostasis, regulating their own levels of certain things, and they have to be able to evolve. So when I say list the basic properties of cells, this is what I reasonably expect. If I ask you to describe the basic properties of cells, you need to go into more detail. So our first cell property is that cells are highly complex and organized. Cellular processes are very carefully regulated. If you remember from your intro biology class, there are some terms like ribosomes and nucleus and mitochondria and Golgi and lysosomes and okay, yeah. You're having flashbacks? Good. Complex. Got it. Check. You can see this even in this figure. So starting from the middle large inset one that they're showing, they are showing you the surface of an intestinal epithelial cell. This is part of the small intestinal wall, which you see pictured up there at the top in the blue. And they give you kind of this zoomed out version, and you can see at the tip of the cell the microvilli along the surface, and they give you an enlargement of that over an inset 2, which is to the left. You can see the finger-like projections that they're always talking about. Inset 4 actually shows you the mitochondria, which is clearly visible, although you can barely see it in the large image of looking at the whole overall cell. If you zoom in even farther on the mitochondria itself, you can see that even in the mitochondria there's complex structure. That's in set 5. In set 6 you can zoom in even closer on one of those tree-shaped things. And in set 7 is the molecular structure of that tree-shaped thing. And over on in set 3, you've got part of the molecular structure of this going on the left-hand side again. So there's all of this going on within a single cell. And that is huge, and it's a vast little world in there. It's a universe all on its own, and that's what we get to explore this semester. So I think it's kind of fun. There are a lot of different levels of organization. You've got, chemicals, cells, tissues, organs, organ systems, organisms, etc. The stuff that you learned way back in, I don't even remember what grade anymore. But we're really going to be focusing on how a cell functions, how our very tiniest little living bits and pieces do what they do. We're not going to look as much at tissues or organs, but we will talk about those at least a little bit. We'll cover little bits about the bladder and the brain and the blood-brain barrier, so that stuff is coming considerably later this semester. Our second property of cells is that they have to have some sort of genetic program and some sort of mechanism for using it. This is basically the central dogma of biology. DNA makes RNA, makes protein. Yep, you've heard it before, you'll hear it again, we'll be going through it again, because it's really fundamental. And all cells that we have found today use DNA as their primary nucleic acid. We kind of think that maybe RNA might have been ancestral, but we're not really sure yet. So it's DNA that is the molecule that encodes the instructions that we need to build everything. Think about that for a second. Think about what you know about DNA. ATGC. Really pretty simple, 4 letters. And yet we have eyelashes and retinas and hair and skin and hearts and lungs and bladders and toenails and all of these different things. How does our DNA accomplish that? It's actually, that's the genetics end of things, but it's still pretty miraculous if you think about it. I try not to use that word a lot, but it'll probably come out. So in addition to just having the DNA, it's how we use that DNA. So if you think about it, we all start from a single fertilized cell, a zygote. That one cell splits and divides into 2 daughter cells that are identical. And those continue to split and divide and split and divide and split and divide. So essentially we are all big clusters of identical cells. But we're not, because the cells that make my fingernails are clearly different than the cells that make my eyelashes, and those are different than my heart cells. They're all the same in terms of the DNA sequence. So what makes them different? That's something that we will explore a little bit this semester. We will look at some of the genes involved in causing us to develop certain ways in certain tissues. Not in a lot of detail, that's for developmental biology class. We'll talk a little bit about epigenetics, which is a relatively new field of biology, and genetics and cell biology and all of that mess. What epigenetics is really all about is little chemical modifications to your DNA. They don't change the sequence. It'll still be ATGC or whatever it was supposed to be, but they change how your cell uses it. These are the little tags that say, don't use this gene, but do use this gene. So this is part of it. Those can change throughout your lifetime in response to environmental things like stress, like taking up exercise, your diet, whether or not you go through a famine. There's a lot. To give you an example of how some of that works, if you were to go back to your grandmother or your great-grandmother's generation, assume you had Jewish ancestry. Even if you didn't, just bear with me for a second. So in the concentration camps, food was very scarce, and because of that, the epigenetics, this little programming all over the DNA, got changed in the women who survived that. Their bodies had to physically alter their metabolism to enable them to survive those very, very low food, those starvation conditions. Unfortunately, those changes are also inheritable. And this is where things get really, really bizarre. So what happened to your grandmother or your great-grandmother, those epigenetic changes can actually be passed down from mother to child over the next 3 to 5 generations. That seems to be about how long they last. They are not permanent, they're temporary. And part of that is because our environment is always shifting and changing. So it may be that for a lot of us, part of the reason we're seeing some of the issues with obesity is because of epigenetic programming. And there's a lot of research going on there to try to figure out If that's true, if so, what can we do about it? Can we develop medications that can change that? Is there some way that we can reprogram our epigenetics to make us lean, fat-burning machines again so that we, don't have to buy size 22 pants or whatever it may be? That's part of it. The other part of it is the junk DNA. Okay, we used to call it the junk DNA. These refer to the introns, the spaces between your DNA that don't actively code for proteins. We used to call it junk because we didn't know what it did. However, in about 2014, we actually finished Project ENCODE, which was this huge effort to sequence those regions, all of those introns in the human genome, and start trying to figure out what the heck they are there for. Because we assume that if it's that much of our genome, it should be doing something. We have found out that a lot of those regions, subsequent to the publication of the actual data itself, we're finding out more and more about the functions of those, and they seem to be involved in controlling gene expression. And that is something that we will delve into this semester. So you'll get to find out, for example, exactly what insulin does to the genes in your body, how cortisol from stress causes you to reprogram something like 30% or more of the genes in your body, and what that may mean. Again, messy. I kind of like messy. I hope you do too, because you're in for a lot of it. Our third cell property is that cells need to be capable of producing more of themselves. Basic cell division or reproduction. Keep in mind, cell division and reproduction are not necessarily the same thing. At a cellular level, technically, sort of, but when you get to the organism level, not as much. My cells divide, that does not mean I have offspring. For that, I actually have to have sexual reproduction. However, some organisms do reproduce solely by the process of cell division, binary fission, like in bacteria. They don't have to have a partner, they don't have to exchange genetics. So cells reproduce via mitosis or binary fission in the case of bacteria, and in this case, each of the daughter cells receives a complete set of genetic instructions. This process is used over and over and over again until you grow up into a nice adult human being, where you continue to use it to replace injured and worn out bits of your body. This initial zygote is formed when a sperm and an egg get together and do the fertilization thing, and what you're actually looking at in this image is an egg cell, an ovum, and you can actually see a polar body that is pointed to by that arrow. This is another, it should have been an egg cell, but it didn't get enough of the cytoplasm and stuff, so it usually shrivels up and dies. Our fourth cell property goes back to metabolism and how cells acquire and utilize energy. And most of it starts with photosynthesis. That's the basis for our whole food chain. Again, intro biology concepts. Photosynthesis provides the fuel for almost all living organisms, even for us heterotrophs that go back and eat the autotrophs. So animal cells, we get our energy from eating the plant cells that get it from the sun. There are some oddballs out there. Chemosynthesis, where some cell types can actually use chemical reactions to provide energy as a source instead of light. That actually happens. There are some deep sea thermal vent creatures that are bizarre that we don't understand well yet. But mostly the things that we deal with, their ultimate energy comes from the sun, gets converted into glucose through photosynthesis, and then gets eaten by other stuff. So glucose is going to be a fairly large topic over these next couple chapters. It is the building block for most of our carbohydrates and our starches. We'll look at that too. You'll get to know more about carbs than you ever wanted to know. And then ultimately we use glycolysis to break that down into ATP, which is kind of our cellular energy currency. Okay, it's your Visa debit card. You can reload it, you can spend it. You reload it again, you spend it again. ATP gets recycled over and over and over and over. So those two molecules are going to be the two big ones for these next few chapters, and basically for most of your cells. Glucose and ATP. This is kind of where it's at. Our fifth cell property is being able to carry out a variety of chemical reactions. Now, this figure is not the one from your text, but that's because I needed something that was graphically a little bit different for this slide. But it accomplishes the same basic idea. So what you see are some of the biochemical pathways that deal with the food that we eat, starting with, up there at the top, glucose, and you'll see ATP and glycolysis and pyruvate. These are words that you remember and are probably inspiring some fear. We're going to work through those. I know. Breathe. It'll be okay. Stay with me. If you look over on the right-hand side of the figure, you'll see that you have lipids and fatty acids, and those feed into your metabolism too. Over on the left, so do amino acids that come from the proteins that we eat. So all of this feeds into our metabolism, and this is a really complex set of reactions, and it's all kind of mixed together in that big squiggly greenish yellow thing that is the mitochondrion. Okay, except for the glycolysis part. That's outside. So some of these, just to give you a very brief idea of what's going on. We've got photosynthesis, glycolysis, the Krebs cycle, mechanisms of carbohydrate metabolism, including fermentation, which makes beer. We've got proteins and enzymes, things like facial peels and baby foot peel, which we'll talk about later this semester, how papaya and pineapple can act as meat tenderizers. This is all based on proteins and enzymes. We'll talk about lipids and fats and cholesterol and which ones are better for your health. RNA is another one of those lovely chemical molecules of life that goes through a surprising number of reactions that we get to look at. How cells communicate with each other is all driven by chemistry. Our immune response, more chemistry. Toxin production, chemistry. There's a lot that goes into this chemical reactions mess, and all of this is going on in something that is microscopic and invisible to the naked eye. How does this even work? That's what we get to look at this semester. Sixth property of cells, they get to engage in mechanical activities. That means they physically do stuff, like intracellular transport, which I'm hoping we get to with the chapter 9 stuff, muscle contraction, which we will be looking at late this semester, and amoeboid movement. And just to put this in perspective for you, I've actually got a couple short video clips. What you're looking at here is a rabbit heart muscle on the top and an amoeba on the bottom. And over the next couple slides, there's actually video footage of these cells moving. And what's really cool about that is that you can actually see at a single cell level that one heart cell contract like a heartbeat. And you can watch the amoeba hunt for food. The seventh criteria for life is that cells have to be able to sense and respond to stimuli, input. And there's a huge variety of potential inputs. This is not even an exhaustive list. We're going to be looking at this a lot through the semester, but cells can migrate away from predators. Many of them, including your skin cells, can follow nutrient concentration gradients, so they can follow a chemical trail of glucose, for example. A lot of them have the ability to respond to light, even if they're not photosynthetic. So what you're looking at there is a euglena, and that dark spot in the middle that you see there is the eye spot, which is actually kind of neat. As it turns out, even our fibroblasts, our skin cells, have the ability to sense and respond to light. People who are congenitally blind from birth can often tell light versus dark, even though their eyes don't function. It's not always the case. Some are more sensitive than others, but we're trying to figure out, how do our skin cells know that there's light? Why would they be able to detect that? And one of the articles that I read a few years ago when I was working in dermatology is that some of the genes that are active in your retinas are actually expressed in your fibroblast cells, rendering them light sensitive, which is kind of cool. Cells can respond to touch. They can respond to different surfaces they're on. They have to be able to sense when other cells go missing. If you get a cut, like, oh, hey, we should heal that and maybe grow some new skin there. So some of this is driven by simple cell contact. And here's a term for you that I did not know until graduate school. Everybody used this word, confluent, and nobody ever defined it for me, so that bugged me. What confluent actually means is that the cells have grown completely into your petri dish until they're all packed together, they're all touching each other, and there aren't any spaces between the cells. So you can estimate how confluent a layer of cells is, like it's 50%, meaning it looks like about half the surface of the petri dish is covered, or 100%, meaning it's completely covered, it's completely confluent, and this is usually when you harvest your cells to go do your experiment. A lot of cells use that touch with other cells as a signal to tell them when to stop dividing. Cancer cells get around that. That's one of the things that we kind of get to look at a little bit this semester too, is some of those signals. So this next slide that you get to look at is actually a short video clip that looks at a fibroblast responding to an infrared light source. So what I want you to look at is the fact that in this slide this video that you're going to be watching, there is a cell that starts off and it's kind of crawling around, and there's a metal pin in its way between it and the pulsating infrared light source. The cell actually has to navigate around this obstacle to get to that pulsating light source. We don't even know why they're attracted to light, but they are. So we're still trying to figure this out. But it's really cool, and there's actually a fair number of videos of cells migrating on YouTube if you need more examples. Our next property of cells, number 8, as if this hasn't been complicated enough, has to do with self-regulation. And this is really important. Cells have to be able to do this, even like multicellular organisms have to be able to regulate themselves. They have to maintain things like ion concentrations, membrane potentials. These are things we'll be exploring this semester in more detail too. DNA and protein levels have to be maintained at consistent balances and levels, and all kinds of really specific control of really minute chemical reactions, which is a pain. They have to be able to sense when to divide, when to go through mitosis. They have to be able to detect DNA damage, or they risk becoming cancer cells. This is critically important during embryonic development as you go from being one cell to being two cells to being a complex multicellular thing. So what you're looking at in this particular image is actually intended to be a simple, really easy experiment that you could pretty much kind of do at home. This is sea urchin development. In the normal pathway, you start off with your fertilized egg, it splits, it becomes two identical cells, those go on and they become basically these little almost amoeba-like looking things. Your little sea urchin there that grows up as an adult. In the experiment, what the researchers actually did was when it reached that 2-cell stage, they split the embryo in half, and those 2 new embryos continued to develop normally and made 2 identical twin sea urchins. This is actually the same process that you get identical twins with for humans. So what's also weird is that while it's still at this 2-stage developmental spot, there's a lot that we understand, because we know that if you separate the cells at that stage, they'll go on to create full, complete, whole individuals. In fact, if you remove a cell and you take it off for genetic testing, the individual will just regrow that cell and no big deal. A baby's not born missing fingers or eyeballs or something because you removed a cell. In fact, this is what we do when we're doing like IVF and the embryos get put through genetic testing. At the blastula stage, when they're still fairly small, just a little cluster, hollow ball of cells, what they do is they go in with a needle and they suck out a few cells and they send those off for sequencing or genetic analysis to make sure those embryos are genetically healthy. And this is a mechanism that we use for screening to make sure that we reduce the risk of birth defects and other major health issues. So it's actually surprisingly common. Finally, our last property of cells. Yay, we got here! And that's the ability to evolve. We have a whole course on this. It's called evolution. Most of you have probably already taken it. So this should come as no huge surprise. The last universal common ancestor is the prototype cell. It was the original cell that had to have its really basic minimal components. What do you think that has to be? What do all cells have in common? What had to be present in that last universal common ancestor cell? Well, all cells have DNA, they all have cytoplasm, they all have a membrane. Not all of them have mitochondria, and not all of them have cell walls. So some of that stuff might have been there and it might not. But at a really basic guess, we can go with membrane, DNA, probably some proteins, cytoplasm, really basic stuff here. Bacteria aren't primitive. We tend to think of them that way because they're small and they're unicellular, but they've got some really amazing complex mechanisms for dealing with things like antibiotics, and we'll look at that shortly. Eukaryotic cells and prokaryotic cells both have changed a lot over time. They have become more complex. Endosymbiosis has led to cells with more internal organelles. In addition to mitochondria and chloroplasts, we've got things like apicoplasts, which have to do with malaria. We only find those in malaria specifically. And this is something that we think actually happened after the engulfing of mitochondria and or chloroplasts. Lateral gene transfer, the ability to zap. Look, you've got some of my DNA. This is also called horizontal gene transfer. It's the ability to transmit DNA from individual to individual, but without having to go through the normal processes of reproduction. Shared genetics, like direct contact, like I could shake your hand and you could get my red-haired gene. Not that you necessarily want it. So all of these may have helped to shape early life on Earth, and it continues to shift and change, or we're pretty sure it does. We do know that there are changes going on today, some of them at the tissue levels in humans, but even some at the cellular level. So there are things that are continuing to shift and change and become different over time. At this point, we get to start talking about the two major different classes of cells, and those are simply prokaryotic cells and eukaryotic cells. I know you already are familiar with those concepts, but it bears some review because invariably every semester I get people who try to tell me that bacteria have a nucleus, and they don't. So, mostly what sets these two types of cells apart is the size and type of their organelles. It's not even that bacteria don't have any internal organelles. They can. They still have ribosomes. They just lack internal membrane-bound organelles of the type that we're thinking, like mitochondria, chloroplasts, a lot of the Golgi type organelles and ER type organelles that are made of layers of membrane that we'll get to a little bit later this term. So the original life forms that started somewhere around 3.7 billion years ago were probably prokaryotic. We suspect that they most likely lacked those sophisticated internal structures that we're more used to seeing in eukaryotic systems today. Again, partially because that's what fossil evidence suggests, and partially because it's simpler, so that's kind of what makes sense. The development of complex life actually started much more recently with the shelly invertebrates over in the Paleozoic, as you're looking at the clock there. This includes the protist animals, plants, and fungi, so we are surprisingly barely even a sliver on that clock of life. Most of the history of life on this planet has had unicellular life, bacteria, algae, that's pretty much it. So all of us complex multicellular life forms, that's kind of a new thing, and I guess we're still kind of seeing how that works out for us. There are some distinct similarities between prokaryotic and eukaryotic cells. They both have a cell membrane that's made out of phospholipids. We'll be talking about the structure of phospholipid membranes in some detail this semester. They both use DNA, and they also have RNA and similar coding structures. They use the same kinds of mechanisms for transfer RNA and ribosomal RNA. We'll be looking at similarities in those systems as well as we work through that material. The transcription and translation mechanisms also similar but not quite identical. In eukaryotes, it's a little bit more complicated, so we'll get into those details as well. Even bacteria can do glycolysis and, to some extent, the Krebs cycle. You don't necessarily have to have a mitochondrion for that. That's just where it happens in eukaryotic systems. So in prokaryotic cells, it can happen on other membranes, like little folded in invaginations on the plasma membrane of the bacteria or the cell membrane of the bacteria. The use of ATP is pretty much universal. Photosynthesis in plants and cyanobacteria occurs in much the same way. Making and inserting membrane proteins is done pretty universally along the same types of pathways, and so is recycling and degrading broken, old, used up proteins. So the metabolism has some really basic similarities, and if you were to try to figure out what might have been present in some of those last universal common ancestor type species, these are functions that we estimate should have probably been there because they are so universal. And it just kind of makes sense that they would have been some of the first things that probably were really kind of cemented into life as we know it today. There are also some really noticeable differences between prokaryotic and eukaryotic cells. The first of those is obviously complexity. And I say obviously because when you look at pictures of them, it should be pretty evident. Prokaryotes are pretty simple. Eukaryotes are way more complicated. We'll be looking at diagrams of cells. Yay! I know you're excited. Isn't that fun? The cytoplasm is actually surprisingly different between prokaryotic and eukaryotic cells. Part of that really has to do with the membrane-bound organelles, whether they're present or absent, and some of the cytoskeletal proteins as well. The ribosomes are a little bit different between bacteria and eukaryotic cells too. Eukaryotes have slightly larger and more complex ribosomes, and later on we'll look at how those are even made. The mechanisms of cell division differ between prokaryotes and eukaryotes. Eukaryotic cells divide by mitosis. Prokaryotes have simple binary fission. Locomotion, again, there's some similarities and some differences here too. Eukaryotes use both cytoplasmic streaming, so movement of the cytoplasm in a directed manner from inside the cell to extend a pseudopod, a little false foot out like you saw in the amoeba video. They also may have cilia and flagella. Prokaryotes can have flagella too, but the structure of their flagella is very, very different than what we see on like a sperm. So they're actually much simpler and much more stripped down, I guess you could say, is one way of looking at it. The genetic material, again, they both use DNA, but how that's handled is different. Prokaryotes don't have a nucleus, so they just have their DNA loose in the cytoplasm. It's usually kind of shoved over into one little special corner of the cytoplasm where they try to keep it sort of protected, and we call that the nucleoid region, as in like a nucleus. Eukaryotes have a membrane-bound nucleus that offers better protection against some of the horrible chemistry that goes on in the cytoplasm. Eukaryotes also have a lot more DNA, and we organize it differently. We put ours in chromosomes that are pretty much linear, and we've got all these histone proteins, and there's all this winding that goes on, and in prokaryotes, it's usually a single large chromosome that's circular, so it's actually sealed on both ends. That actually is kind of cool because it protects it from degradation. There's nowhere for enzymes to start really chewing on the DNA because there is no end piece hanging off. However, prokaryotes like bacteria and some of the archaea also may have little circular plasmids, little loops of DNA that carry some specific genes, like for drug resistance. This is particularly common. So those may or may not integrate into the full chromosome at some point. It depends on what they are. It depends on whether environmental conditions favor that. So they're not really big enough to be considered fully separate chromosomes, though. They just don't have enough genes for that. So just to give you a fairly reasonable, good overview slide and kind of a side-by-side comparison, this is what we're looking at. There are a few things that I've already covered on this, so I'm not going to reiterate those, but I do want to point out that some of these actually do have some other differences. Like there are actual differences in the cytoskeleton. In prokaryotes, they have a much simpler cytoskeleton, and in eukaryotes, it's more complex. The cell wall in prokaryotes may also be different. It's actually made of different stuff. So if you're looking at a bacteria, its cell wall is going to be made of peptidoglycan. If you're looking at a plant, it's more likely to be cellulose. If you're looking at a fungus, it's more likely to be chitin or some other type of structure. So you can actually identify different types of cells based on what their cell walls, if present, are made out of. If it doesn't have a cell wall, generally that's an animal cell. In bacteria, you also get conjugation pili. These are used for exchanging plasmids. So this is a mechanism to share genes and do the horizontal gene transfer thing. It is not actually for reproduction. I kind of was mistaught some of that myself early in my biology career. And they're not useful for motion. They're not for swimming. They're just kind of like these little tubes that you transmit DNA through. Prokaryotes are generally haploid. They only have one copy of the chromosome, and again, most of the time their reproduction is asexual, so simple binary fission. Eukaryotes, on the other hand, have more complicated structures. They are more likely to be diploid, so have 2 or more copies of each chromosome, and their whole system for cell division mitosis is more complex. Yes, I know, still relatively simple: prophase, metaphase, anaphase, telophase, but that's a lot more than what goes on during just simple binary fission. We also have structures like centrioles to help us organize all that, so there's quite a bit more that goes into eukaryotic cells. Here we have Joebacterium. This is pretty much the kind of prototypical structure of a bacterium. So starting from the inside out, we have the big blue squiggly DNA in the nucleoid region. They didn't really shove it over into one corner, but it doesn't really matter. You see dark blue dots kind of floating around in there. Those are the ribosomes. You've also got some other little bubbles of stuff floating around here and there that aren't particularly labeled. They can still form vesicles. It's not as extensive as what we see in eukaryotes. Moving more toward the outside of the cell, the innermost layer of this system along the inside is the plasma membrane. Same structure, phospholipid bilayer, all that happy stuff. Next, on the next layer, this blue-greenish layer in here, this is actually part of the cell wall. And this is the part where you would actually have the peptidoglycan, since we're looking at bacteria. Outside of that, you may have another layer of membrane in some cases. You may also have a mucus-like capsule that helps to allow the bacteria to stick to surfaces, like your nasal epithelium. Or you can have no capsule. It varies from species to species. We do have a bacterial flagellum pictured here, and we have several, many conjugation pili. Again, remember, these things are not involved in reproduction. They are just there for exchange of DNA, so lateral or horizontal gene transfer. And of course, the fluid-filled portion is still called the cytoplasm. Yay! So this is about as simple as it gets, and this is why we usually start with bacteria, because they're relatively simple. Next up is Joe plant cell. I call all my cells Joe. There's a reason for that. My high school biology teacher named all her cells Joe, so therefore all my cells are still named Joe, because, well, that's my little homage to my high school biology teacher. She was a little strange. She wanted to name her children things like Annabina. Look it up. So back to our plant cell here. The first thing that should be obvious is that this is way, way more complicated than the bacterium. So again, starting from sort of the inside and working our way out, we have a nucleus in this case in the top left corner of the cell, where again doesn't make a huge difference. This is just where they pictured it. Inside the nucleus, you've even got a nucleolus. We will be talking about that in more detail this semester. You finally get to learn what the thing is for. It's not just a big dark spot. There's things that go on in there. The nucleus is wrapped in a double layer of membrane, and that outer layer is actually continuous with the rough endoplasmic reticulum that you can see branching off of it there and covered with ribosomes, which is what makes it rough. Let's see, other than that, I guess I'll work my way down the left-hand side. You've got a Golgi and some mitochondria. Yes, plants have mitochondria too. People somehow seem to forget that. Bunches of ribosomes, those, again, dark bluish dots. Vesicles, cytoplasm, cytosol is the watery part of that. That giant other blue bubbly thing is the central vacuole, which is responsible for storing water and ions and degraded bits of proteins and things. And that's what gives your plants turgor pressure. So When they have plenty of water, that will fill up and it will swell and it will push against the cell wall and make your plant all perky and standing up straight and everything. As the water levels in the plant drop, this thing will shrink and your plant will gradually wilt and then die because you forgot to water it. Yeah, water your plants when you get home. Let's see, above that we've got peroxisomes, smooth endoplasmic reticulum, which does not have ribosomes all over it, chloroplasts, there's microtubules over there on the right-hand side as well, so a lot more stuff going on in there. Again, in this particular case, you still have a plasma membrane that lines the inner surface, and the cell wall is actually secreted outside of the plasma membrane. So the components for the cell wall, since this is a plant, that's cellulose, get pumped through the plasma membrane and they assemble on the outside of the cell wall. Or outside the plasma membrane to form the cell wall, I suppose I should say. Animal cells, also much more complicated than bacteria. They are structurally very, very similar to plant cells with a couple major exceptions. We don't have a central vacuole. We don't have a cell wall. Most of the rest of the stuff we still have. We also may have a few structures like centrioles that are not particularly common or seen in plants. We also do things like make glycogen granules. We'll talk about the differences between glycogen and starch in the next chapter. But a lot of these things are still the same. Ribosomes, Golgi, nucleolus, cytosol, nucleus, etc. So most of your internal structures are basically the same. One of the things that I would encourage you to do through this semester is build yourself a cell concept map. We're going to be starting with basic molecules, but as we start looking at things like the nucleus, draw yourself a sketch of the cell. It doesn't even have to be pretty. Write out the stuff that you know about what goes on in there. Draw little sketches of things and label them, and build your own map like this in your own little scribbly handwriting to help you remember what goes on, because you will be able to go into way more detail than this slide shows by the end of the semester. I guarantee it. To look at some of the different types of eukaryotic cells, we've got a vast array of different structures that can be present. This is a cell from the male rat reproductive tract, obviously, so that's the testes. Yay, exciting. I'm sure this is something you've always wanted to know. So my first question for you is, what is that thing? What does it look like to you? Yeah, I know, there's a label on the diagram. Ignore it for a second. What is your first thought? Egg? Blob? That's okay. These things over here, hot dogs, blobs, I don't care. I'm not all obsessed with food. My point is this, when I look at some of these pictures, I still see blobs too. It's okay if what you see is a blob. You do have to learn eventually to recognize some of those blobs as being things. So the big thing with the dark spot in the middle of it, that's, yeah, probably the nucleus, and that dark spot is probably the nucleolus. In this particular case, you've got some chromatin shoved off over here in the corner of your nucleus, which is actually kind of cool. One of the things that we talked about in the last slide with the animal cell is that the rough endoplasmic reticulum is actually continuous with the outer layer of the cell membrane. So that's what they're trying to indicate in here. And it's very hard to see this in electron microscopy pictures because it's really, really super thin. I mean, it's only phospholipid bilayer thickness, so, you know, itty bitty. Smaller than that, but itty bitty. And so this kind of surrounds and branches off from the nuclear membrane. Again, hard to see. It tends to show up as slightly wavy lines. As you move out into the cell, you're more likely to encounter things like smooth endoplasmic reticulum and the Golgi complex. They've got one of those. They're saying that this thing in here is a Golgi complex. I guess I'll have to take their word for it because I can't see that much difference between that spot and that spot, which I'm guessing is another Golgi. And this thing over here, which actually does look more like a Golgi that I tend to think of with the flattened out discs and stuff. That looks like a Golgi to me. But it takes practice to be able to spot those things. And then you've got all these other things. The dark black spots. The other granule type spots. The, what is that thing? Things. It's not critical. It's okay. They're blobs. Unless you do a lot of electron microscopy, you're not expected to be an expert in it. But you should be able to reasonably identify what the structure is kind of pointing at and go, that looks pretty much like a nucleus. So that's what I wanted you to get out of that. The cytoplasm, you guys are taught that it's mostly water. Yep, it's mostly water. But on the other hand, look at that. That is a mess. That is a big mess. The elements of the cytoskeleton are actually stained with a dye that makes them red in this case. The ribosomes show up in green. Okay, that's the light blue tealish stuff all over in here. I don't think that's green. That doesn't look green to me, but I'm not perfect with like color vision. The cell membrane out here at this edge is the stuff that they're saying is blue, and yes, that I would actually believe is blue. So what goes on in the cytoplasm isn't just water and salts. There's a whole lot of other stuff that's free floating in there. It's more like a meshy, soupy mix, and that's probably still not even an adequate comparison. But it gets the point across. This is a much busier space than we think of it being because we're mostly taught that it's water. So we kind of envision, you know, water, soup, tea. I don't know what you mentally picture cytoplasm as looking like. Whereas the reality looks kind of more like ramen. I promise to stop with the food comparisons eventually. Cell reproduction also varied between prokaryotes and eukaryotes, so we actually get to see a little bit of the differences here. One of them is reproduction on the left. What you're looking at is a cell going through mitosis, and in particular, in this case, this is some beautiful fluorescence microscopy work. The green star-shaped things, those are parts of the centrioles. Those are the microtubules that emanate out of the centrioles, and they attach to the chromosomes, which are stained in blue. So using our lovely knowledge of mitosis that I'm sure you all remember so very, very well, does this look like prophase? No, it's too organized for that. Metaphase, better. During metaphase, the chromosomes should line up in the middle of the cell. That's pretty midline. Anaphase, they should be pulling apart toward opposite ends of the cell. They're still pretty close to the middle at this point. It doesn't look like they've really started to separate well, so I'm probably going to have to say that's closer to metaphase than anything else. So, nice review for you there. And just a really pretty picture. I love fluorescence microscopy. Every year Nikon actually sponsors what they call their Small World competition, where researchers and scientists and people take their fluorescence microscopy images and they submit them, and you get to vote on them, and the prettiest 12 end up in a calendar for the next year. So it's actually a lot of fun. Yes, I have voted on them in the past. Over there on the right hand side, what you see is bacterial conjugation. I was told that this is like the bacterial version of sex. It's not. It's not actually for reproductive purposes. I've said that a few times by now. I'm still trying to drill it into my own head in some ways. So what you see is this F pilus, the conjugation pilus, that extends from one bacterium all the way up to the other bacterium. And if I were to actually show you this image and ask you which one's the donor and which one's the recipient, I wouldn't necessarily expect you to figure that out. The only way I can tell you which is because when I look at the two bacterium, one of them is clearly covered with lots and lots and lots of conjugation pili sticking out all over the surface. It's very fuzzy, this one down here on the bottom. And the other one does not have all the conjugation pili. So we call the one with the pili the donor. The other one's the recipient. And the only thing that's going to happen is that DNA, like those little plasmids, little fragments of genes, they're actually going to unwind and decircularize, and they're going to loop their way through this F pilus, and they're going to be transmitted up to the recipient bacterium up here at the top, where they should reform a nice little circular loop and plasmid, or they could integrate into the host chromosome. Either way. Chapter 1 is long. But we get to talk about sperm next. So, If you remember, I said that there were differences between the structures of sperm of flagella of prokaryotes and the flagella of eukaryotes. And this is where you actually get to see some of those differences. So, for example, over here on the left, what you're looking at is a bacterium that is covered with lots of flagella. They don't just have to have one, they can have many flagella. And this usually enables them to swim better. The more flagella you have, the faster you can move, the more likely you are to outcompete your fellow bacteria. When you look at the structure of the bacterial flagella under electron microscopy, as you zoom in that inset, you can see that it's actually not all that much to see. It's really just kind of like a tube with a little collar thing on it and another little thing and another little thing. That's it. There's nothing really to it. If you look at a eukaryotic flagella, we actually have this very particular arrangement. It's called usually the 9+2 arrangement. And that's because you have 9 clusters of these tubules going around the outer part of the sperm, and you have these 2 microtubules on the inside part of the sperm. These are connected to each other by these Dynean arms. As these arms contract, they cause a ratcheting motion. They pull on those pairs along the outer surface, and it causes the flagellum to twist a little bit. That twist creates a spinning motion, and that's what pushes the sperm forward. So now you know how sperm swim. Some of the birth related issues that can lead to infertility actually arise because of breaks or malformations in those Dynean arms, and that's one of the major reasons for infertility in men. Just in case you were really, really curious. You'll learn more about sperm this semester too. They're actually one of the first cells that was discovered under the microscope because of their relatively large size, so people have been kind of fascinated with them for a while, and we're still actually learning a lot about them. Prokaryotic cells also have a surprising diversity. There's actually 2 large domains of prokaryotic cell types. The archaea, the weird ones, the ones that like live in the deep sea vents, and several years ago, it was, let's see, what year was it? 2010. This article was published in Science magazine, one of the big journals, and it actually said that they found these bacteria that lived in a lake in California that had extremely high levels of arsenic. Now, what do you know about arsenic? Yeah, it's on the periodic table, and it's poisonous. So researchers were really, really surprised. And they isolated some of these bacteria, they took them back to the lab, and this article was actually published, and it said that they actually used arsenic instead of phosphorus in the DNA. Now, chemically, if you go pull up a periodic table, go ahead, that's what Google's for. Pull it up in another window, take a quick peek. Arsenic should fall directly under phosphorus in the periodic table. So find the P, look immediately under that, and you'll see arsenic, which means they've got the same number of electrons in their outer shell. Yay, chemistry! Okay, that means chemically they react very similarly. The only real difference is the size of the atom itself and how many protons it has in the nucleus, which is also related to the physical size of the atom. So they thought that these bacteria had the capability of substituting arsenic into their DNA instead of phosphorus. And everybody was so excited about this, including the researchers, and they actually proposed that it might be a whole separate tree of life. Unfortunately, I read the article, and some of the techniques that they used weren't totally convincing. For example, there was one thing I really, really wanted to see from them that they had not done, and I wanted them to isolate the DNA from these bacteria, complete with the supposed arsenic in it, and run it through a mass spec. Just a standard old mass spectrometer like the one Abby uses on NCIS, and if there's actually arsenic in the DNA, you should get a nice peek for arsenic. They didn't do that experiment. I wasn't convinced, and I wasn't the only one who had a problem with that research. In fact, I think the paper was retracted. So it's kind of sad, but really they overstated their case. It's still really interesting because somehow these bacteria were living in extremely high levels of arsenic, They were probably archaea, and that's fascinating, but it fell way short of a whole separate tree of life. The bacteria are the stuff that we're actually probably way more familiar with, like mycoplasma, which are the smallest known bacteria. These actually become a huge problem in terms of scientific research. Mycoplasma can actually get into the cell. They are an intracellular infection, so they get inside the host cell and they'll live and replicate inside the host cell, a lot like viruses do. They're hard to detect, and you have to have special screening kits to do this, and it's almost impossible because it doesn't respond well to antibiotic treatment. So if your cell cultures get a mycoplasma infection, you probably won't know. If you're getting them screened regularly and you find them with mycoplasma, the best option is really just to go order new cells. And yeah, there are actually places you can do that. Other types of bacteria include the cyanobacteria, the earliest cells that we think probably did photosynthesis. These gave us ultimately the organelle that we now call a chloroplast. Very good. It helped to accumulate oxygen in our atmosphere. Some bacteria can do other chemical reactions. What you're looking at on the left there is a cyanobacterium in electron micrograph image. So that's an electron microscopy picture. And you can see all these wiggly, wavy lines that look like layers and layers and layers of membrane. Keep in mind, they don't have intracellular organelles. They don't have complete little bubbles of stuff in the same way that eukaryotic cells do. But they can make layers of plasma membrane, and they can fold that in like what you see going on in some of these structures. They're just little blebs, they're just little bubbles, but they can use that to sort of enclose some stuff and sort of sequester some stuff. This gives them space to do some chemistry separately from other chemistry. We'll see why some of that's important as we work through the semester. What's really cool is the cyanobacteria that you can actually find growing on polar bears. Green polar bears. That is awesome. Polar bears are supposed to be white. I see all the Coke commercials at the holidays. Polar bears are white, except when they grow cyanobacteria in their fur. There's been some evidence that there may be versions of cyanobacteria that are growing on the exterior surfaces of the International Space Station. I haven't read any follow-ups on that, but a couple years ago they came out with an announcement that they found stuff living on the outside of the ISS, you know, in space, with no air, with no oxygen, with vacuum and no life supportiness that we currently think of. So, that's interesting. Don't know quite what to make of that. We're not sure how that happened. There are way more types of prokaryotes actually in the world than there are anything else. So we usually do this based on DNA sequencing today. Again, as our technology has improved, we don't just look at pictures and go, that looks like a prokaryote and that looks like a eukaryote. Now we actually need better evidence, and we can get that from DNA sequences. We are finding out that there are lots of them in the oceanic subsurface and the terrestrial subsurface, so just below the surface of the soil in both, you know, dry land and under the ocean. Soil itself actually has about 26 times 10 to the 29th cells. That 10 to the 29th, okay, Avogadro's number. 6.02 times 10 to the 23rd. This is times 10 to the 29th. Oh dear God, that's a lot. And if you actually look at how much carbon that causes, or how much carbon is actually used by those prokaryotic cells, it accounts for something like 26 times 10 to the 15th grams of carbon. That's a fair amount of weight that's just bacteria in the dirt, and even more in the oceanic and terrestrial subsurfaces. So if you look at the overall number of bacteria in the world, you are looking at numbers that are absolutely incomprehensible. Most people can't even really comprehend having $1 million, much less a million of anything. And we're dealing with, that's 10 to the 6th. We're dealing with 10 to the 23rd higher than that. Yay. This accounts for a fair amount of the biomass of our planet. There's actually more bacteria in and on your body than there are human cells. They outnumber our human cells. So essentially, we're giant walking colonies of bacteria. Some of you now want to go dip yourselves in bleach. Please don't do that. 1, it will burn. 2, it's actually not good for you. 3, you've got tons of actually useful, beneficial bacteria that you really, really need. 4, bleach has a pH of like 13 or 14. Not good for your skin. So where did these eukaryotic cells come from? How did we get from prokaryotic cells to eukaryotic cells? The idea of endosymbiosis has been with us for a while now. So what we think happened is that one prokaryotic cell engulfed another type of cell, and in essence, instead of digesting it, it was like, oh, hey, wait, you're making food for me. This is an awesome relationship. You can stay. Hi, look, I've got a pet. And thus we got mitochondria. And then somewhere later down the line, this happened again, and that cell, now that had already one invader, got another one that could do photosynthesis, and ooh, look, we got a plant cell. That's part of it. What you're looking at in the image there is actually another bacterium with complex internal membrane structures. So you can actually see that they don't have classic organelles like what we learn about, but they do actually have loops and bubbles and invaginations of their membrane that start to look like the beginnings of organelles. So the idea that something like this cell shown here might have become one of the early mitochondria becomes a little more plausible. We have additional evidence for that. So some of that evidence comes from the physical structure of these organelles. Mitochondria and chloroplasts both have a double membrane layer. So they've got one layer and then they've got another membrane layer, and this suggests that they actually invaded a host cell and got completely engulfed by it. Typically that actually results in a layer of membrane getting brought into the cell. We'll actually look at that process later this semester too. I know, you're tired of hearing later this semester. We'll get there, I promise. Patience, patience. Both chloroplasts and mitochondria also have their own DNA, and sequence-wise, it's distinct from that of the host. So you can actually sequence the mitochondrial DNA. We hear about this even in crime shows today, so it's fairly well known. But chloroplasts have their own DNA, and it turns out that if we sequence that, what we get for chloroplasts looks like the modern genome of cyanobacteria. In fact, it falls right between two different groups of cyanobacteria, and it's fairly closely related to one of the algae C paradoxa. So, photosynthetic, related to cyanobacteria, very plausible. The mitochondrial DNA looks like proteobacteria, and again, it sort of maps to groups of proteobacteria. What's also interesting about this is that the chloroplasts and the mitochondria both can go through binary division in the cell without the host cell reproducing. So the host cell doesn't have to go through mitosis to get more mitochondria or chloroplasts. They can divide on their own. This is one of the things that happens when you do aerobic conditioning when you work out. As you progressively train your muscles, as you place more oxygen demand on them, you are going to actually trigger replication of your mitochondria. And it's only temporary. So it'll last for a few weeks, maybe a couple months or so. And if you're not using all those extra mitochondria, they'll gradually dissipate back down to baseline levels. However, if you keep in conditioned fit state, then you retain those mitochondria and larger mitochondria and more copies of them, and you get more efficient workouts with less soreness after, which is totally awesome. I hate running. This idea of 3 kingdoms is surprisingly newer than you think. And I mentioned that I began college in 1996. This still was not being published in textbooks when I began my undergraduate studies in 1996. However, the data that led us to this decision goes all the way back to 1977, which is a year before I was born. What this work that you're looking at here is they took comparisons of the 18S, the small ribosomal subunit, and they compared this across different species. And they looked at yeast, Saccharomyces, they looked at L cells, they looked at Lemna minor. Those are all eukaryotic cell types. They looked at E. coli, Colobrium vibriiforme, Bacillus firmus, Corynebacterium diphtheriae, chloroplast DNA sequences, methanogens, methanogen. Okay, lots of methanogens. Those are the archaea group. And what they found was they were looking for sequence similarity. And look at the numbers. They're not huge. 33% similar, 29% similar, 36% similar. Okay, there's a lot of variety there. But look at what happens when you compare these outside of their group. If you compare Saccharomyces cerevisiae to E. coli, their genomes are actually only about 5% similar. Way lower than the 33, 36-ish. And in fact, if you look at this whole cluster, you can see that none of these numbers are at all similar to what we were seeing within this group. We see the same kind of thing when we come down to the next group that's highlighted in the reddish, pinkish, whatever you want to call that color, and you look at the bacteria. Their genomes within this group are more similar to each other than they are to anything else. They don't look like the eukaryotic cells, and they don't look like the methanogens, the archaea. Same thing when you look at the methanogens. Their genomes are pretty similar to each other and very, very different from everything else. So this is the first evidence that we saw that indicated that we really ought to consider something above the kingdom level, and that's where domains came from. In fact, when I started college in 1996 in my biology for undergraduates class, we were still arguing over whether this should be included in textbooks. The data was pretty convincing, and today you guys were probably taught from a very young age that there are 3 domains of life, but it wasn't always so. I grew up with just kingdom, phylum, class, order, family, genus, and species. Today we've got domain, kingdom, subkingdom, superphylum, etc. It's gotten more complicated. We're revising this based on what we find out about gene sequences and ribosomal DNA sequences like what we're looking at here. And that's cool. We should revise it as we get better data. This is more detailed and it's more quantitative. It's more numerical. It's harder to argue with. It's not based on something looks like something else, but they're really fundamentally different. This is looking at the most fundamental stuff about an organism. So that's how we got here to our 3 domains of life with bacteria, archaea, and eukarya. The bacteria include the ones that we tend to think of, like strep and staph, both the gram-positive and gram-negative types of bacteria, and the cyanobacteria. The archaea include the halophiles and the thermophiles and the methanogens and all the weird stuff and the acidophiles and the things that live in absolutely bizarre climates, and probably those arsenic bacteria too. The eukarya is the stuff that most people are more interested in, including plants, animals, fungi, protists, like the euglena with the eye spot that we saw earlier. Okay, nifty. So where did eukarya come from? How did we get from being prokaryotes to eukaryotes? The idea of endosymbiosis is awesome and all that. You know, that gives us the mitochondria and the chloroplast, but our genome itself is still different. And this is kind of part of an interesting story. So when you look at our DNA, it actually suggests that maybe we're sort of a fusion of an archaea and a eubacteria, a regular true bacteria. Some of our nuclear DNA is more closely related to the archaea, and some of our organelle DNA looks like it's more eubacterial. So we also see these cases of gene transfer, which gets even stranger. There are actually genes that leak from inside your mitochondria into your nucleus on a fairly regular basis. We've actually observed the same thing with genes from chloroplasts that can leak into the nucleus. And that's what this diagram is actually all about. They took a gene for antibiotic resistance, neo-STLS2, and it's shown in yellow, this little yellow squiggle in there, over here in this particular chloroplast. So there's our yellow squiggle. And this gene is inactive when it's in the chloroplast, so for the gene to be functional, it has to be incorporated into the nuclear DNA. So you're waiting to see this transfer happen where the gene jumps from the chloroplast into the nucleus. That's a case of lateral or horizontal gene transfer. Once this occurs, the gene can become active and it can function, and those cells become immune to kanamycin, for example, one of the antibiotics. So that's all they had to do, was treat these plant cells with really high levels of antibiotic and look for the survivors. And it turns out that this happened in every 1 out of about 16,000 cells. That is way more frequent than we expected it to be. This is a process that continues, it's ongoing, it's even going on in your body right now somewhere. Some little gene probably just escaped from a mitochondria and got incorporated into your nucleus. Humans typically have somewhere between 300 and 600 mitochondrial DNA genes that have leaked, that have bled from the mitochondria into our nuclear DNA. It varies from person to person, and yet we still function fine. We've even scrounged bits of viral DNA and bacterial DNA from intracellular infections like mycoplasma, and we can find bits and pieces and remnants of those infections in our own DNA, which is really, really weird. So, what does that mean we are? I guess we're a fusion, we're a hybrid. Kind of a strange thought. To kind of delve into this a little bit more, if you look at the RNA, enzymes, and ribosomes, the gene sequences for those all show that they are actually related, but our genes involved in transcription and translation seem to be the ones that came from the archaea, while a lot of our other genes that are related to making proteins seem to have come from bacteria. So even our own genetics suggest that we are a fusion product of, on the one hand, a eubacteria, and on the other hand, an archaea. So eukaryotes, really, based on everything we've got so far, look like they're probably a fusion of 2 other domains of life, which is totally radical. People wonder why this hasn't happened since, and there's actually evidence that it has. If you look at malaria, Plasmodium falciparum, they actually have a third organelle called the apicoplast that is not present in any other organism. And what it looks like happened there is a eukaryotic cell engulfed a plant cell. Now, there's something kind of fun for you. And if you look at the genetics of those apicoplasts in the malaria parasites, what you actually find are still little remnants and bits and pieces of genes for photosynthesis. How cool is that? We're hoping that maybe, eventually, maybe we can actually use that to target the malaria parasite and develop a better treatment for malaria that doesn't make the host sick too. But it's not like malaria goes out and recharges in the sun. Those genes are really degraded, they're really broken up, and they don't appear to be functional anymore. Sad. There's a lot of specialization in eukaryotic cells too, and we've already kind of touched on this because we know that our heart cells are different than our fingernail growing cells. This is why you don't have fingernails growing out of your chest cavity. Glad I could give you nightmares. Wasn't that nice of me? So what you're looking at here is just a very complex but still single-celled organism, Vorticella. And it's got this weird spiral, almost spring-like contractile part of the stalk, a giant nucleus in this thing, giant nucleus in this thing that's indicated by the arrow. It's got multiple copies of its genes. Plants actually are, and even some unicellular eukaryotes, can actually be triploid, tetraploid, polyploid, pick a number. I'm not sure how many copies Vorticella actually has. If you're that curious, you can go look that one up. I haven't been that curious. Cell specialization becomes really important when we're looking at human tissues in particular. So this goes back to a question that I basically had in 7th grade. I didn't know how to ask my teacher because I was a kid, you know. But we just learned about mitosis and meiosis, and I understood that. But what I didn't understand was how I went from being one cell, it divides by mitosis, you get 2 identical cells, those divide, why am I not just a big rolling blob of cells? Shouldn't I be just a big ball of identical stuff? I didn't know how to phrase the question to ask my teacher, why do I have nerve cells and red blood cells and smooth muscle cells and fat and intestines and striated muscle and bone and other connective tissue? Where did these things come from? If they all start from the identical same precursor, how do I get all this tissue differentiation? We're learning some of that is due to epigenetics. We'll talk about some of this a little bit during the semester, but again, that's really more the realm of developmental biology. But in multicellular organisms, this becomes a critical part of how we function. We kind of divide up different areas of specialization within our body. The numbers and arrangements of organelles even differs. Your heart muscle has a different number of mitochondria than your skin tissue does. Your skeletal muscles have different numbers than your brain tissue, your neurons do. And this actually kind of makes sense. These cells are metabolically active. You know, my eyelash growing cells, nowhere near as much. One has a lot of need for mitochondria, the other has very little need. So we do see differences in there. Despite that, there's still a lot of stuff in common, which is why most of the time we're going to talk about pretty much generic cells. A lot of our studies have come from several model organisms. So these are the most commonly studied organisms that we have in biology today. There are reasons for that. Most of them are fairly easy to grow or raise. Bacteria like E. coli can be grown in, well, okay, it's E. coli, pretty much anything. Saccharomyces cerevisiae, that's yeast. We use this at home to make beer, to bake bread. It is very quick to replicate. It is the simplest eukaryotic thing that we usually work with. Arabidopsis is a very simple plant. The C. elegans nematode is the simplest animal that we usually work with. Drosophila fruit flies, those have been foundational for our studies of genetics, which you probably already know. And Mus musculus, the mouse. Yeah, okay, that's a naked mouse. It doesn't have any fur. Actually, in the lab, if you have nude mice, they are usually severely immunocompromised, and you would never, ever, ever hold them in your bare hand like that because they're going to die. In fact, if you have to handle nude mice in the lab, you usually have to put on a face mask and a hairnet and gloves and more gloves and booties and a smock or something like that, and you have to scrub down with at least alcohol before you can handle them, and then after you handle them and before you touch another mouse. They can be a pain. So that image made me kind of angry in the first edition of the textbook that I used, and they haven't swapped it out yet despite my complaining, because I don't think it's fair that they killed a mouse just to take a picture. Maybe it wasn't an immunocompromised one, but that's generally the assumption. So these are our model organisms, and the one thing that we do have on these is very, very complete genomes. We have fully gene sequenced all of these organisms at this point, so we know everything that we can know about their DNA, but we still are learning a lot about how they develop. E. coli is pretty easy, but it's actually possible to break E. coli. Trust me, I've actually done it. Yeast, we're still learning about how they process and make different kinds of fats and carbohydrates and stuff. We're still learning about gene regulation in plants and animals. So all these studies are still ongoing. Most of our model organisms are chosen because they've got relatively quick generation times and they're easy to grow or take care of. Mice, for example, they're kind of well known for breeding. I mean, I know the saying is to reproduce like bunny rabbits, but really, bunny rabbits don't have anything on mice. To put this in perspective, for those of you who are curious about mouses, a mouse, from the time it is born, it typically takes about 3 weeks of nursing. It's usually weaned around week 3 or so, and around week 6 to 8 it becomes sexually active. At that point in time, you can get your female mousie pregnant. They are pregnant for about 6 weeks. So basically about every 6 weeks, you can have more and more and more and more mice. A typical litter of mice is somewhere around, I'm going to call it about 6 based on my experience. The highest number that I've seen that survived was 15. So that's lovely. On top of that, as if that wasn't fun enough, mice have this wonderful thing called postpartum estrus. So as soon as the mommy mouse is finished delivering her babies, she goes into heat. She can get pregnant again the same day, within a few minutes to a couple hours after giving birth, back-to-back pregnancy. And you wonder where all the mice come from. Yeah. So, like I said, quick, relatively easy to breed. Yes. This brings us to other topics because we are actually looking at human cells and human tissues too, and we are in particular really interested in the idea of stem cells right now. So stem cells are undifferentiated. They're just generic cells that have the capability to turn into a variety of other types of cells. We're hoping that what we can do is learn to take these stem cells, harvest them, and use them to grow new tissue for damaged or diseased adult tissue. So like, for example, you have a heart attack. Cool, we give you an injection of stem cells into your heart, and instead of ending up with scarring, you get nice new heart cells. Maybe we could learn to do this for the brain and avoid some of the problems with Alzheimer's and Parkinson's. We could potentially work with this for the pancreas, for people whose pancreatic beta cells fail early in life and they develop type 1 diabetes. Maybe we can actually restore function to some of these organs that is lost. So it's not all about being mad scientists and trying to get X-ray vision, and yes, some of us secretly do still hope for that kind of thing, or not even secretly. But we hope that we can actually use these to find treatments or even cures for human diseases that do affect us. So what's kind of cool about these cells is that most of them, like I said, they're undifferentiated, they're generic, and the best time to catch those is during early embryonic development, when you've still basically just gone from a zygote to bloop 2, 4, 8, 16-ish cells. You don't want to get much bigger than that because that's really when differentiation sort of starts, is when you get around the gastrula size. So you have to harvest these fairly early. And this brings up ethical issues like, when does life begin? That's not a discussion that we necessarily have to have in class, but it is not uncommon for us to take one of those cells. When we looked at the sea urchins, we already saw this, and to use it for genetic testing to make sure that the embryos that we do choose to implant are healthy and free of genetic diseases. What if you could take those embryonic stem cells and freeze them, and then later in your life, if you do have a heart attack, you've got your own cells that you could use? Heck yeah, I want that. Unfortunately, as we grow up, our tissues mature and we get fewer and fewer stem cells in our adult bodies. We still have stem cells, but they're not the same as embryonic stem cells. Embryonic stem cells, especially in this early stage, are totipotent. T-O-T-I-P-O-T-E-N-T. Totipotent. And what this means is that they can actually develop into all the different kinds of cells and tissues in your body. As you get older, as you start to develop distinct lung tissue and brain tissue and all that, even then you've still got some stem cells in your lung, you've still got stem cells in your brain. Even as an adult, you've got stem cells at the base of your little hair follicles on your arm in your skin. These are important because they help you heal after injury. But they're already partially differentiated. They can't become, for example, brain cells again, or not easily. So those are called pluripotent stem cells. One of the things that we know of that we are encouraging is banking of cord blood, and that's because embryonic cord blood, when you take the umbilical cord at birth, it has a large proportion of embryonic stem cells. So the thought is, if we harvest those, if we freeze those cells then if the child actually develops a childhood leukemia or something, we can actually regenerate their bone marrow with their own healthy cells. There is no risk of rejection. Unfortunately, that's really, really expensive still. So how do we use adult cells, like the ones in our skin, at the base of our hair follicles, how do we get those to go back and become embryonic cells? That's tougher than we thought it was going to be. Initially, we thought it was going to be really easy, like suck nucleus out, stick it in an egg, and watch life happen. And that's actually pretty much kind of how we do things for cloning today, and that's partially how we generate stem cells, but they are induced stem cells. So we take an egg cell, an oocyte, you still have to have an egg cell, you suck its nucleus out, whee, bye-bye, and you take a stem cell from a regular cell, you take its nucleus out and you transplant it into your now empty egg. Once you do that, it should sense that, oh, hey, I'm an egg, I've got 2 sets of chromosomes, I must have been fertilized. Time to replicate. Bloop, bloop, bloop. And it should start to grow and divide. Sometimes it takes a little bit of chemistry magic or a little electrical shock to kind of kickstart that process, but this induces your embryonic stem cells to start growing. We can grow them in culture, and we're finding out that we can even get them to differentiate if we control the stuff like the food substances that we put in the media properly and the hormones that we put in the media. But we're in the dark ages of how to do that. We're not even in the infancy of that as far as I'm concerned. So if you were to do this, if you were to take a bunch of cells like this and you were to do this, you can also separate when they're still in that blastocyst stage. You can separate those individually like we did with the sea urchin, and you can get lots and lots of little clones to develop. And that's how we do cloning too. Isn't that fun? When you meet my kitty, I will talk to you about why I'm thinking about having her cloned. It'll cost me about $50,000, which is an insane amount of money. And it's not just that I love the little fluff ball, it's for science. The ability to take cells and deprogram them and then reprogram them is something that we are really only very, very beginning to tentatively scratch the surface with. So we know that we can, for example, take skin cells from a mouse, put them in culture, treat them with certain types of transcription factors. We'll talk about what those do later this semester. They basically control gene expression, and we can get them to turn into basically pluripotent, maybe not totipotent, but at least pluripotent stem cells. We have been able to do this in culture. We can even do some genetic corrections with these. So again, there's hope that maybe we can reverse diabetes, type 1 diabetes. Maybe we can reverse other genetic disorders where things go badly wrong. This would be awesome. And that's what the real hope is for stem cell therapy. A lot of people do have ethical concerns about where these stem cells come from, especially the embryonic derived stem cells. Those, unfortunately, kind of remain our best source because they are still so totipotent. But a lot of the cultures that we've already established have been contaminated with things like mycoplasma that I've mentioned before. So we lose some of our ability to do research with that. They're not normal anymore. They're not untouched, naive little cells. So their development might actually be different because of those infections in the culture. I don't know. I've actually thought about donating eggs or something like that for stem cell research because I'm not using mine and I don't really care. So why not? I think giving people the option to donate unused embryos for scientific purposes might be okay. A lot of them may have genetic disorders, and if we can study those, maybe we can find other ways to correct them in children who aren't conceived through IVF, natural-born children. There's, again, still a lot of ethical concerns. So that's something that we do have to think about and that we do have to address. Moving on a little bit, we're going to kind of change tracks here for a little while, and we're going to look at some of the other aspects of cells and their biology. Cells are usually measured in terms of micrometers, that's 10 to the minus 6th meters, and nanometers, 10 to the minus 9. And that's tiny. In fact, it's generally pretty much just below the threshold of what humans can see with the naked eye. So kind of as we start down there at the bottom of the slide and work our way toward the top of this figure, we're looking at the sizes of stuff that you can normally see. Frog eggs are typically about, they say 2.5 millimeters in diameter, which is funny because there used to be frogs in my pond and their eggs were pretty good size. They were actually closer to a centimeter in diameter. And that's way bigger than 2.5 millimeters, which is really tiny. A paramecium is 1.5 millimeters long. You can see that on a ruler. That's not even too horribly difficult. However, when you get into the 200 micrometer and below range, 0.2 millimeters and less, that's where you really need a microscope. You can't see stuff. Our eye just isn't good enough to resolve that, to be able to see that. Our biggest cells are things like epithelial cells. These tend to be up to around 30 micrometers in height. The fibroblasts in your skin, I've personally measured them. On average, they are 20 to 25 micrometers across in diameter, so they're fairly large. Your white blood cells, they're considered big, and they're 12, so they're about half the size of a skin cell. Things you can also see with a basic light microscope, you get down into the chloroplast and mitochondria. Those are fairly good sized. You can see those easily. Bacteria get trickier. They can be as little as 1 micrometer in length. And cilia is right at the very edge, down at the 250 nanometer range. Those are really hard to spot with a standard light microscope, and I have never forgotten my first time seeing them. I was in genetics in college. I finally had a microscope where the lenses weren't all scratched up and stuff, so I actually saw cilia for the first time, and I actually yelled out in class, "Holy shit!" I was really, really thrilled. Pardon the language, but true. I'd never actually physically seen cilia before myself. All I'd seen were diagrams. So when I could finally make out those teeny tiny little hair-like things coming off the surface of the cell, that was really, really exciting to me. And I still think it's exciting. However, that's as small as it gets. As you get below that threshold, you need something better than a light microscope. This is where you need electron microscopes. This is for stuff like viruses. Ribosomes aren't visible without electron microscopes. The structure of the plasma membrane, which is only 5 nanometers wide, again, you need an electron microscope for this stuff. So this actually limits how big cells actually can get. And a lot of it actually does have to do with diffusion through that lipid bilayer. You can tell I'm really good at drawing with a mouse. Things have to be able to diffuse through this freely, and that's kind of a slow process. Even though it's only 5 nanometers, the chemistry of that doesn't let a lot of stuff through easily. We'll go over that more in the next chapter. But from there, it actually also has to be able to diffuse from the surface of the cell all the way into the middle of the cell to really be able to evenly distribute nutrients and stuff. This is what limits cell size, is how long it takes for substances to really diffuse through the cytoplasm. If you get much bigger than this, it really becomes very inefficient and you end up with areas of the cell that are starved, and that just doesn't work. So, be familiar with basically what you can and can't see with a basic light microscope when you need to switch to an electron microscope. Hint, viruses. You don't actually have to memorize the size of HIV. I'm not going to be asking you questions that are that nitpicky. I just want you to have a good handle on what you can see with your eye, what you need a microscope for, and when you should switch over to an electron microscope. We've been working on synthetic biology, and this is a field, it's also sometimes called biohacking. This is the attempt for us to play God. We want to try to create a living cell in the lab. This is probably scaring some of you. You're going, we are not responsible enough for this. And sometimes I agree with you. But we want to try to explore what are the minimum genes that we need to make an organism function? What are the minimum things that have to be there to make a cell? Because this may help us understand the origins of life. It may help us understand what the first life forms looked like. It could even help us identify life on other planets, which, yeah, okay, I get excited. So the toolkit that we actually need really needs stuff like nucleic acids, proteins, fats, lipids, other types of biomolecules. But we have maybe applications to medicine or industry or the environment. What if we could take a bacteria and engineer it to clean up oil spills or other chemical spills? What if we could engineer something that could deal with radiation? This could be really cool. What if we can come up with inks that write themselves? What if we could come up with, I mean, the world of what if is out there for biology too. So what biohacking is all about is taking the genetic tools that we've developed and playing with it, experimenting with a sort of not quite no holds barred sort of a sense. You can actually order things like these bio bricks that I'm kind of showing on this slide. What this is a little gene sequence that come in little modules. So you're like, I need a gene that turns on with glucose. Fine, this is the part of the gene that turns on for glucose. Then I want to tie that onto another part of a gene from a different gene that's going to make a blue glowy thing. And all of a sudden, you've now got a blue glowy thing that turns blue and glows when glucose is present. So you could use this to maybe make a little patch of skin that could glow blue and tell you if you need insulin if your blood sugar levels get too high. How cool would that be? No more pricking your finger. On the other hand, kids would probably play with that too much and try to glow in the dark. That could be bad. But this kind of synthetic biology, these kinds of like little bits of Lego-like things for your DNA, they're available. There's actually a biohacking lab here in Hampton Roads. It's on Granby Street downtown. It's run by Jameson Duncan, and he's a really cool guy. But there are some minor restrictions on what you can do with this. For example, you are not allowed to work with biowarfare agents or pathogens, specific pathogens. So nothing that is going to make people sick, nothing that is going to be dangerous. The point of biohacking is to try to improve our understanding of biology and medicine and to let people who have the out-of-the-box ideas go play. Because my brain's all corrupted. I went through a traditional PhD. There are people out there who are way more imaginative and way more creative than I am, who may have an idea and maybe they can get it to work. I might tell them it's impossible, but I could be wrong. So why not try? Why not do the experiment? We've already made some strides in this. And the first synthetic life form was actually created a little while ago now, and it's been termed Cynthia for synthetic life form, and oh how cute. It was based on Mycoplasma mycoides, a specific strain of this, and this first sort of synthetic life form, what they actually did was they took these cells and they actually sucked out their own host genome. They designed a genome on the computer based on the natural gene sequence, but with some differences. They added some genes and stuff. And this was 1 billion base pairs long, so 1 billion ATGC letters. They had this custom made in the lab, which we can do, and they injected that back into these DNA-free mycoplasma and got them to start dividing. And it kept dividing successfully. So these were our first synthetic cells, where the genome was actually created by humans. This was actually initially started by Dr. Craig Venter. He founded the Human Genome Sequencing Center, and he's actually one of the people that's responsible for inventing modern gene sequencing as it is today. When we started the Human Genome Project way back in the 90s and stuff, they thought that it was going to take like 30 plus years to complete it. What Dr. Venter did was he came up with ways to let computers do a lot of the work for us, and it's way, way faster. He actually gave a talk over at the Ted Constant Center several years ago in the Big Blue Room, and I can't find a single reference to it anywhere but I was there, so I know it happened. He was actually talking about personalized medicine. And since then, this is one of the projects that he's gone on and done, is looking at how to make synthetic life. From here, now that we can actually do some of this and manipulate genomes like this, we can start programming these kinds of cells to, I don't know, make antibacterial products, to make insulin, to make proteins that we can use for correcting other issues in the body, to grow new skin or tissue types. I mean, theoretically, the options are limitless, and that's what the hope is for this field. The last part of chapter 1 is thankfully a little bit shorter, and this actually is where we get to look at viruses to some extent. First thing, obviously in big, large text there, viruses are not cells. They are generally not even considered to be alive because they don't have most of the characteristics of cells. They don't have their own metabolism, and while they're outside of a host cell, they don't have mechanisms for glycolysis. They can't move, they can't do much of anything except kind of sit there. I personally think of them like a suitcase for genetic material. It just sits there like a bomb until somebody opens it. So they are classed as pathogens because they can cause disease. They are obligate intracellular parasites. They have to get into a host cell to replicate, to function, to take over the host cell, to cause disease. All of that cannot happen without a host. Outside of a host, technically they're called virions. So what we usually have is genetic material, which can be single-stranded or double-stranded. It can be DNA or RNA. So you can even have double-stranded RNA, which is a thing that's not supposed to exist. And this is usually surrounded by some sort of protein capsid, which is what these diagrams are trying to show. For some viruses, outside of that, there's also a lipid envelope that would be around the outside surface of this, which would also help this to escape immune detection, bind to cells, and enter cells. So not much to them in many cases. Some of them have additional proteins packaged inside them. So like HIV, for example, which we'll look at in the next slide. That actually has some additional proteins stuck in there too, like reverse transcriptase, that comes prepackaged with the nucleic acid. So many of our viroids are pathogens, like the ones that cause cold and flu. Those are viruses. They actually use RNA as their genome, not DNA. And they typically cause problems for us because when they get into our cells, they kind of co-opt our whole translation machinery, the ribosomes and stuff, and our cells get so busy making viruses that they forget to do normal cell functions, and that's why they become dangerous. So there's a lot of different types of viruses. There's a lot of different shapes of viruses. And Dr. Barexi, who teaches genetics, also has a phage discovery course. And what you do is you go out and you collect like samples from the environment and you try to isolate viruses that are just found in the dirt. They're out there. Some of them actually infect bacteria, like the T-even bacteriophage there on the right. You'll notice that it's got a pretty sophisticated structure, but even there's not really that much to it. So you've got your nucleic acid, which in this case is RNA, the red squiggly thing. You've got kind of a shell around that that's made up of some proteins. You've got an additional layer of proteins and some lipid bilayer that came from whatever host cell birthed it. And you've got some little surface proteins called GP120 that usually allow this to bind to other cells. That's how they get into cells in many cases, is they'll bind to a receptor and the cell will suck it in thinking it's food. That's it. That's a virion. That's a virus. The HIV virus looks kind of like a funky spaceship. So what kind of happens is, as this virus gets into your bloodstream, it will dock with certain proteins on the surface of your white blood cells. These little feet will bind to those on the surface of your cell, and then this spring-like thing actually contracts and it shoots your nucleic acid, which in this case is also RNA. It shoots this out into the cell, so it acts almost like a little syringe, and it injects this directly into the cytoplasm of your cell. And that's how it productively infects your white blood cells. The adenovirus structure is even simpler. So there's a huge amount of diversity. There's a huge amount of different ways you can make viruses. They have different shapes. They have different styles. Some are more complicated than others. This one, you'll notice the T-even bacteriophage. It's even got reverse transcriptase, which is kind of in little itty bitty dots in here, which are very hard to see. But there's a few of them in there. So a lot of how viruses work, again, they bind to those surface receptors. These are usually fairly tissue specific. For example, flu, influenza. There's no such thing as the stomach flu. Stop it! Okay, rant aside. What happens is the receptors on your cells are very specific for certain kinds of cells. So your nasal epithelium has certain types of these receptors sticking out, waiting to catch food or bacteria or whatever. The viruses exploit those, but the flu virus, for example, is only good at binding to your nasal epithelium. It doesn't usually productively infect your digestive tract. It almost can't. There are a few rare cases and a few strains that can and do, but we tend to think of this kind of as a lock and key mechanism. And we'll be revisiting this when we talk about enzymes too. So the same idea will come back to haunt us a fair amount. What you're looking at in this particular set of images are two different sort of patterns of viral infection. So there are really kind of two life cycles for a virus. They can take option 1 or option 2. Some of them don't really have an option. But a lytic infection is the one that we think of most often. And what happens during this is the virus gets into the cell, it replicates and it replicates and it replicates, and it buds out from the surface of the cell. And eventually the cell is pumping out so many viruses that the cell bursts and it lyses and it dies. And that's why it's called a lytic infection. The other life cycle for viruses is sneakier. And this is what HIV does. So in this case, the virus actually gets into your cell. We can use HIV as a model, and that's what you're actually seeing here. And in the early stages, what happens is it actually gets copied backwards from RNA into DNA through that reverse transcriptase. And then that little bit of DNA that codes for HIV gets incorporated into your genetics. Horizontal gene transfer from a virus. This is one perfect example of it. So it becomes part of your cell's DNA forever and ever, and its offspring's, amen. Which is part of why it's been so hard to find a cure for HIV. How are you supposed to cure your own immune system? It's now part of you. It's part of your body. Throughout your life, sometimes the virus will reactivate and you will bud some viruses from the surface of your cells. Those can go on and infect other white blood cells, and since it's an infection, your whole immune system gets revved up, producing lots of targets. Problems. Yeah. So those are our two life cycles, lytic and integration. And those are our two main models. The host, sometimes this can even cause cancer, because when those genes from the virus incorporate into your DNA, it really depends on where they insert. What if it inserts into a critical gene for cell should not divide, and your cell just starts dividing wildly? Voila, tumor. This is part of what happens, and part of how, like HPV, papillomavirus, this is thought to be part of how it actually becomes malignant and carcinogenic. So if all they really are is just little packages of DNA or RNA, they don't have any metabolism of their own, they don't really do much of anything on their own, what are they? There's three current main ideas on what viruses might actually be, and there's still a fair amount of debate because the evidence is really mixed. Some people think that these represent escaped genes, so these selfish genetic elements that replicate within a host genome, just like a computer virus. Actually, computer viruses were named for human viruses. And that at some point, these selfish genes actually just kind of accumulated enough extra genes through horizontal transfer to have enough proteins to allow them to exist for short periods of time outside of host cells. Colds and flu, for example. Colds can actually persist on surfaces for a few hours. Flu for a few hours. Some viruses can persist for days on surfaces, and some can only last a couple minutes. Another idea for where viruses came from, or what they actually are, is the reduction hypothesis. And this is a little different. It's the idea that these were parasites. A parasite's normally dependent on its host, just like a virus. And the idea is that over time, the viruses actually lost genes and lost genes and lost genes that weren't critical until they got to the point where they actually just became totally dependent on the host, the ultimate parasite. And that actually makes some sense. We actually see gene leakage from the mitochondria into our DNA, so why not? This is plausible. And the third idea that we have going is that they may be relics of an RNA world, a time before DNA, that these might sort of represent some sort of pre-cellular life. Since we find viruses that have both DNA and RNA as their genome, this actually starts to look more and more plausible. In fact, if you trace back the lineages, RNA viruses are older than DNA viruses. Their genomes converge farther back in time. That was one of the scientific writing assignments we actually looked at one semester a year or so ago. I don't remember how long ago anymore. But it was interesting because they actually showed some comparisons between the viruses that infect different types of organisms, and they found out that the viruses that infect bacteria, their genomes are actually different than the viruses that infect eukaryotes, which kind of makes some sense. Bacteria are different than us, so the conditions for survival should be different too. Which of these is correct? We may never know. Could it be all three? Actually, I kind of lean in that direction. I think all of these are potential reasonable explanations. And I think that maybe there's some variation as to exactly how some of those viruses came about, but in general, yeah. So what do you do for viral infections? Stay home, drink your fluids, rest, chicken soup. There's something weird about chicken soup. They've actually conducted scientific research, and they actually have found that chicken soup works better than a lot of medications. While we're on the topic of flu, the flu test is only 50% accurate. Seriously, look it up. They actually told me that in the doctor's office a couple years ago. They tested me for flu. It came back negative. They said, we're going to prescribe Tamiflu. And I said, but my test was negative. And they said, it's only 50% accurate. To which I go, then why did you test me? If you're going to ignore the results of the test, and if it's only 50% accurate, I can flip a coin. I'm not paying for a $30 or $50 flu test that's only as accurate as a coin toss. Give me the Tamiflu anyway. So be aware of that. Tamiflu interferes with viral replication. It doesn't do much against bacteria, so we've developed antiviral drugs too. They exist differently. They are much harder to target, and our ability to treat viral infections is still pretty limited compared to what we can do for bacterial infections. Anyway, that's the end of chapter 1. I hope you found it interesting. I know it was long. It takes me a while to go through it in person too, but it should give you a lot of thought starters for this semester. Next, we will be delving into the biochemistry of cells.